Foot and mouth disease is still one of the worst plagues of livestock around the globe. In large parts of the world, the disease is still pretty much rampant. It's not under proper control. There's always the chance of the disease leaking from there into places like the UK where it's normally free. To try and control it at the moment, there's something of the order of 3 or 4 billion doses of vaccine given every year. But it doesn't work quite. In places like India, they've got a very good program of vaccination, but they can't meet the needs because they can't make enough vaccine. One of the major problems is that you have to produce the vaccine from live, highly infectious virus, which always carries a risk with it. We're building the technology to make completely safe particles. Essentially, they're just the components of the virus that the immune system needs to see to mount a response. We've shown that you can protect cattle with these particles. We're just publishing a paper now where we describe using diamond, using knowledge of the detailed structure to re-engineer the particle so that we can make it stronger and hence make a more stable vaccine. The production of the particles is based on some very innovative work from Ian Jones at Reading University. They're so small you can't see them with a normal microscope, so you'd probably get 1 billion or so on a pinhead. And yet, using diamond, we can see them at the atomic level, which is amazing. We're now standing on top of the diamond synchrotron, 0.5 kilometre in circumference, and essentially an extra high-powered microscope. A visit here of a few hours, and we can get enough information to determine the atomic structure of a redesigned virus shell, which is a crucial step forward. The preliminary results look very encouraging, yeah. It should make a difference. It should be able to get to market and really start to help animal health on a global scale. We put this together as part of a collaboration with a brilliant group of collaborators, including Reading University and Oxford, led by Brian Charleston at the Pirbright Institute, and have managed to bring together funding from DEFRA and the Wellcome Trust principally. What I hope is that this will be an example of how a synchrotron can enable a much more sort of intelligent approach to redesigning vaccines and getting a next generation of vaccines.